Lysosomes. Introduction. Lysosomes are discovered by the Belgian biochemist Christian de Duve in 1955. Later on, it's reported by the P. Mattei in 1964 in Neurospora, a fungus. The membranous vesicle appears as a tiny bag-like structure with the average diameter of 0.23 to 0.5 μm. They are regarded as a digestive enzyme of the cell or suicidal bag. The term lysosome is derived from the Latin word line, meaning to dissolve or loosen, or soma, meaning body. They are mostly present in animal cells, while only in some lower plant groups. Meristematic tissue of higher plants is also found to be contains lysosomal vesicles. They are found in the most abundant numbers in the cell having secretory functions such as liver cells, pancreatic cells, kidney cells, spleen cells, leukocytes, and macrophages, etc. They are originated from the cisternae of the Golgi body or from the tubules of endoplasmic reticulum. In animals, it is found suspended in the cytoplasm in all cell types except RBC. Structure of lysosomes. They do not have a definite shape. They are pleomorphic. They are mostly globular or granular in appearance. They are 0.5 to 0.2 to 0.5 millimeter in diameter and surrounded by a single lipoprotein membrane in pure composition. The membrane or glycosylated proteins named as lysosomal associated membrane proteins and lysosomal integral membrane proteins. Both lamps are stranded in the inner surface of the membrane, forming a coat layer. Inside the membrane, the organelle contain different hydrolytic enzymes in crystalline form proenzyme. Type of lysosomes. There are two types of lysosome, primary lysosome and secondary lysosome. Primary lysosome, small sac-like structure which contain enzymes synthesized by the through endoplasmic reticulum. It is actually a storage vesicle called as storage granule storing enzymes. Secondary lysosome. It is formed by the fusion of primary lysosome with the endosomes or phagosomes. If therefore contain engulfed particle in addition to the hydrolytic enzyme. Engulfed endocytoside particles are progressively digested by the enzymes. It is the structure of the lysosome. Types of lysosome. There are three characteristic subcellular vesicles that are characterized as a secondary lysosome on the basis of morphological features. These vesicles are following: digestive vacuole. Small sac-like bodies containing foreign materials engulfed by the cell in the structure, digestion, or breaking down of foreign body takes place. Residual bodies. They are secondary body with unhydrolyzed residues due to incomplete indigestion of ingested particles. The unhydrolyzed particles or residues either remain inside the lysosome or discharged out. Autophagic vacuoles. During autolysis, a process in which lysosomes release hydrolysis in damaged or aging cells to digest them. This vacuole is formed known as autophagosomes. Proton transport and activation of the enzyme. The lysosome mainly have H plus ATPase, are also known as vascular ATPase, VATPase, which transport H plus ion inside the lumen. Proton influx and infusion of endosome activates the lysosomal enzyme, which are otherwise inactive. These are the proteolytic enzymes as the cathepsin, collagenases, and elastases, lipolytic enzymes, lipases, phospholipases, fatty acid esterases. For carbohydrate splitting enzymes, alpha glycosidases, beta glycosidases, hyaluronadases, and aryl sulfatases. Nucleic acid and hydrolyzing enzymes, ribonuclease and deoxyribonuclease and other enzymes acid phosphatase and catalase. Formation of the lysosomes. Lysosomes are formed by the fusion of transport vehicles budded from the Golgi network with endosomes, which contain molecules taken up endocytosis at the plasma membrane. The hydrolytic enzyme formed in the rough endoplasmic reticulum are tagged with the mannose-6-phosphate by the enzymes and glycosamine and phosphotransferase and N-acetylglucosamine phosphoglycosidase with the glyco apparatus to target them to the lysosome. The tagged enzyme bud off from the Golgi and nascent vesicle, then bind to the late endosomes, which is another acidic membrane-bound organ. The late endosome has a proton pumps within its membrane that keep its intra-environment acidic. Functions. Lysosomes serve two major functions: intracellular digestion. Primary lysosome fuses with membrane of food vacuole and skid the enzymes to food vacuole site. Digested food then diffuses through the vacuole into the cytosol to use it for the energy and growth. Lytic enzyme action. Cell organelles that are no longer needed are engulfed by the lysosome to form the autophagosome. Cell organelles inside the autophagosome are then degraded by the hydrolytic enzyme of lysosome. Simple molecules after digestion diffuse to the cytosol to use it for the energy and the growth. Heterophagy is the taking up of exogenous particles by the process of phagocytosis to form phagosomes, which later fuse to form primary lysosome to form heterophagosome. Nutrients obtained after digestion of ingested particles in heterophagosomes are released into the cytosol. Autophagy is the process in which the subcellular organelles are taken up the lysosomal vesicles and degraded by the hydrolytic enzyme contained in the lysosome. This process leads to the destruction of the cells in the body, also termed as a cell suicide. Extracellular digestion, where a cell secretes hydrolysis outside by exocytosis, resulting in the degradation of extracellular matrix. Example: cells of the digestion lining, alimentary canal, secretion by the saprophytic enzyme, etc. Autolysis is the process where the killing of an entire set of cells occurs by the breaking down of the lysosomal membrane. The autolysis occurs mainly during the metamorphosis of amphibians and the insects. Fertilization involves the rupture of acrosomal membrane, which results in the release of enzymes on the surface of the egg. Acrosomes are basically a modified form of lysosome, can be termed as a chain lysosome, forming sperm head. The rupture of sperm or acrosomal head paves the way for the entry of the sperm into the egg by digesting the egg membrane. Thank you.